Hello everyone. So in previous lectures we have seen the structure of bacteria and the types of bacteria based on its shapes. So in this lecture we are going to look at the gram positive and gram negative bacteria, which are the difference in which we are differentiating the bacteria on the basis of its cell composition. So if you haven't watched that lecture yet, please watch that lecture so that this lecture will be easy for you to understand. I will give the link of that lectures in the description box. Here gram positive and gram negative bacteria. In 1884 the scientist named Christian Gram, because of that scientist Gram, these bacteria are named as a gram positive and gram negative bacteria. So what he did is he did some staining procedure to differentiate between the bacteria, and the bacteria which retain that stain, the stain that he used, which he called the gram positive bacteria, and the bacteria which did not retain that stain, it is called as a gram negative bacteria. So the procedure of gram staining that we will look in further lectures. So right now we are just knowing that what is the difference between gram positive bacteria and gram negative bacteria, and it is based on the cell wall composition. So we will look at exactly what's the difference between the gram positive bacteria and gram negative bacteria. So in the lecture of a structure of bacteria, we have seen that bacteria contains a cell envelope which consists of a plasma membrane first, then the outer membrane is a cell wall, and then some bacteria have the capsule. So here we are concerned about the plasma membrane and the cell wall. So this is the basic structure of a this is the basic structure of the any kind of bacteria which contains the cytoplasm. Then it is enclosed by this plasma membrane. In both cases we can see the plasma membrane, and then the basic difference, the main difference there, because of which we say one bacteria is gram positive and gram negative, is the peptidoglycan layer. So bacteria mostly consist of the peptidoglycan layer in which cell wall. So both gram positive and gram negative bacteria consist the peptidoglycan layer. But what the difference is, in gram positive bacteria you can see this red portion which I have drawn is peptidoglycan layer, and in this gram negative bacteria this is the red portion that is peptidoglycan layer. So here you can see the difference is this peptidoglycan layer in case of a gram positive bacteria is very much thick as compared to the gram negative bacteria. This is very thin peptidoglycan layer in case of a gram negative bacteria, and in a case of gram positive bacteria it is very thick in layer. So this cell wall contains the cell wall of gram positive bacteria contains a very thick layer of a peptidoglycan, and in case of a gram negative bacteria it consists of a very thin layer of a peptidoglycan layer in this red portion you can see, and above that it consists of some outer membrane which is made up of a lipopolysaccharide layer. So the structure is this is a plasma membrane which is present in both of the case. Then the thick peptidoglycan layer is present in the case of a gram positive bacteria, and a thin peptidoglycan layer is present in a case of a gram negative bacteria, and then there is outer membrane also above this peptidoglycan layer. So this is the difference between gram positive and gram negative bacteria. So what is exactly the function of a cell envelope, this cell wall, cell membrane, or the peptidoglycan layer thickness, the size? What is the major function of this is to provide the stability, provide some rigidity and protection to the bacterial cell so that it should not burst. So if we compare bacterial cell to the other cells like human cell or plant cells, bacterial cell is more susceptible to burst, or they are present in very unpredictable conditions. So it is very important for them to protect their cells from bursting out. So that's why this peptidoglycan layer, thick peptidoglycan layer, helps them to protect from the cell bursting. So that's why this peptidoglycan layer or this cell envelope in all is very important for bacteria to survive. So that's why this is the difference between gram positive and gram negative bacteria. So this extra thick peptidoglycan layer protects bacteria from bursting. So that's the importance of this peptidoglycan layer. So the example, if you can see the gram positive and gram negative bacteria, the examples are I have written over here. The gram positive bacteria is Staphylococcus aureus, and the gram negative bacteria is we have we have always used this bacteria as our culture, or we always play with this bacteria in our laboratories, microbiology laboratories. So the E. coli, which is very familiar name for us, which is a gram-negative bacteria. So this is the basic difference between the gram-positive and gram-negative bacteria, and how we can observe the gram-positive and gram-negative bacteria under a microscope. That we will look in further lecture, where I will explain the procedure of a gram staining. So that's it for today. This is the gram-positive and gram-negative bacteria difference. So here we are differentiating the bacteria on the basis of a cell wall composition. So that's it. And in previous lecture we have seen the differentiation of bacteria on the basis of their shapes, and here it is on the basis of their cell composition. So that's it for today, guys. Thank you for watching my lecture. Please like, share and subscribe. Thank you.